Okay, it's my distinct pleasure to introduce Dr. Gabriel D. Erausquin, who is a physician scientist who's double-boarded in neurology and psychiatry with fellowship training in deep brain stimulation. He currently serves as a distinguished university professor of neurology, psychiatry, and radiological sciences at University of Texas Health Science Center. His research expertise focuses largely on prediction and prevention of severe brain disorders using global health strategies for community engagement, early detection, and early intervention in combination with advanced precision medicine and bioinformatics and cell-based models with a focus on brain development and repair. And at a personal note, it's really a pleasure to work with Dr. Gabriel because he is not only a physician, but he challenges us computer engineers on EEG signal processing, on some of the machine learning models. So his expertise spans across these wide domains, and he has deep understanding of not just the physiology or the neurology and psychiatry, but also on the ML and AI models. So it's an honor to have him here today. Gabe, the floor is yours.
Thank you very much, Derisha. I hope I can live up to all that introduction. That was very, very kind of you. Thank you. But as was already said, I'm a physician, which means that I work with patients. And as any of you who has ever watched an episode of Dr. House knows, what doctors do is driven by this idea of diagnosing problems, making a diagnosis. And in the original Greek form, the term diagnosis means simply to classify, to tell apart, to separate things, which is something that you all are very familiar with. The processes of classification are a big chunk of what computer engineers do in order to develop applications. Now, classification can be a relatively simple problem when the entities that one wants to classify are neatly separated, as is the case of these three colors. But that's rarely the case when one has to translate any class into a real-life measurement. And for instance, even a relatively well-classified set of colors, if one turns that into, say, measurements of a fluorescent emission in a microscope, the distribution of those measurements begins to overlap. However, small the overlap might be, there may be already cases in which both of the properties of, or rather the properties of two classes begin to overlap. As you can make the reality more complex, as you move into situations in which the colors begin to overlap deliberately, such as this, the process of classification becomes increasingly more complicated.
Gabe, I just, sorry to interrupt, but did you mean to share your slides yet or not?
Oh, yes, I thought I was sharing already. My apologies.
Oh, no, sorry, we can't see them.
My apologies. Let me start sharing, if I can do that. Thank you for the interruption. That was timely. Let's see. I thought I was sharing already. My apologies. Do you see my slides now?
Yes, we can.
Okay, so I was saying three neatly defined colors, measurement of those colors and some overlapping in individual cases. And as you begin to move forward into more complex situations in which those colors combine deliberately, the measurements become increasingly overlapping. And if the situation becomes increasingly fuzzy, the measurements are very hard to distinguish, such that the distributions begin to look like a uniform space. So how do we go about separating these sets of wavelengths, or these individual colors, when the reality is fuzzy, as it is in most biological systems? Well, You are all familiar with the idea of multidimensionality. We can start from a process in which there's a single dimension, just like the example that I just gave here, in which all of the distributions appear to be the same. As you move that from one dimension into the second one, and say you add, in addition to the measurement of wavelength of the fluorescence, you add temperature or some other similar measure, the distributions begin to separate slightly. And the more dimensions you add, the more neatly they separate. But also, as you increase the dimensionality of the problem, you narrow the distributions of the individual sets of measurements. Each dimension becomes narrower, but also becomes sparser. In other words, the distributions become more and more separate from each other. This is a sort of a simple example in which you have candies of two different tastes, spicy and sweet, and you want to separate them based on color. And you sort of can tell that there are some reddish and some bluish candies, and the ones that are bluish tend to be sweeter, the ones that are redder tend to be spicier. But if you actually turn that into a more refined measurement, eventually you come into a singularity process such that each of the candies is completely separate from the rest of them based on the particularities of the color. So this is what's being called, as you all know better than I, the cars of dimensionality, meaning that the more dimensions we add, the number of points that you need in order to establish clear classes or clear separations of classes becomes increasingly larger, such that you go from, say, a situation in which you have 3 points, or 3 points are enough to identify a class in a single dimension. As you increase the number of dimensions to, say, 8, not to go too high, you need several thousand points instead of 3. So that is when you are looking at relatively simple problems in classes that are relatively well defined. But biological complexity far exceeds those examples. Let me give you a few instances of what I mean by biological complexity, which are very well, if you will, very well described in the literature, and that paint the picture for those of you who are not of biological background. This is the first example. You're looking at 3 different animals, each of which has wings. Yet the actual structure, and each of them uses the wings for flying. The actual structure of the genetic and protein processes that lead to the creation of those wings is completely different in each case. And the wing in the butterfly is a radically different biological instrument, if you will, or a biological set of processes that lead to the creation of that instrument from the eagle. Conversely, if you look at the upper extremities or anterior appendices of different animals that are all mammals, even though they may look very different and they have very different uses, their underlying structure can be quite similar. So the human hand, a dog's anterior paw, a wing in a bird or a whale have all the same underlying architectural features, even though they are used for very different purposes and have very different appearances. When you look at genetics, the situation becomes increasingly more complex. And this is really a problem that you should all be very well familiar with. These are 2 dogs. Any human can tell that the smaller one is a Chihuahua and the larger one is a Great Dane. Doesn't take a great deal of knowledge of the species to know that these are 2 dogs. Easy classification, easy diagnosis. Yet, there is no available classifier, automatic classifier driven by images that can make the separation accurately. If any program, say you take, I don't know, Alexa, one of those, you ask any classifier to, or Netflix, the Netflix classifier, ask them to classify these 2. If they think the big one is a dog, they will tell you that the smaller one is a rabbit or a rat. If this one is classified as a dog, then this will become a calf or a cow. So this what seems to us a very simple diagnosis problem. It is for automatic image analysis processes incredibly hard. Yet we all know that the genetics underlying these two animals is identical. The problem becomes even more complicated when you talk about humans. And you have identical twins, in this case, these two children, that have different enough features of their brains. In this case, if you look at it, these are magnetic resonance images, for those of you who are not familiar with them. They're taking the coronal plane, meaning that they're sectioning the brain this way, parallel to the face, if you will. And the spaces that are black in the inside of these images that look like a small butterfly are the so-called lateral ventricles that contain fluid. The child that has the smaller lateral ventricles, and I hope you can see the difference, although it's subtle, I'll talk a little bit more about more evident changes, but the butterfly here is a little larger than the butterfly here. You can say that the bottom part of the wings is larger in this case than in this. So likewise, the segment of the so-called third ventricle, which is this fluid space down here, is larger in this child than in that child. Subtle as that might be, the associated behavioral picture of that is that one of the children has autism and the other one does not. As you expand the features of the analysis and say, look at cortical thickness, meaning the thickness of the small band of tissue that overlays the brain, you find that the child that has autism has much thinner cortex in a number of different parts of the brain. So this level of subtle analysis is again an expression, the complexity that comes from interactions between genes, which are identical in those two children as they were identical in these two dogs, and environmental or other developmental dynamic processes that lead to completely different phenotypes over time. So how do we approach this problem? How do we try to dissect that biology in real life? So I mentioned that dimensionality can become increasingly complicated, and we will come back to this when we talk about genetics. But one of the ways in which you can try to understand the complexity is by precisely looking at multiple dimensions, looking at the thickness of the cortex, looking at the size of the fluid spaces inside the brain, looking at the behavior, looking at gene expression, looking at protein quantities or protein levels in different tissues, and so on and so forth. But the more you add to the complexity of the measurement, the larger it is, at least in theory, the number of individuals that you have to look at in order to be able to classify them correctly. And this has been a problem for diagnosticians with which we have struggled mightily. So how do we go about doing this in a quantitative way? The second problem that we have is the problem of locality, which can be well presented in the example of the brains. If I look at these two brains and take as a single measurement the average thickness of the cortex, the average, across all of the parts of the brain, all of this blue tissue, if you will, of the blue parts of the brain will become average with the red parts of the brain. If you do that across individuals that have slightly different parts of the brain that are affected by the process, what you end up with is with an average that will be completely unable to distinguish what parts of the brain are the ones affected. So we have to find out really what are the specific parts of the brain across multiple individuals that are affected locally, not just as a global average. So the way we have approached over the course of the past 10 years or 15 years, these problems is by trying to use clustering methods that fulfill a number of different criteria, if you will. The first one is that we want clustering methods that are fuzzy enough to allow multiple sizes of clusters. We don't want to have a forced clustering such as that we have to prejudge or predetermine the number of clusters we want to find. Second, because that would exclude the possibility of, for instance, identifying these processes, right? So we want multiple clusterings that allow enough fuzziness in the number of identified classes. Second, we want clustering that is unbiased in the sense that, or as unbiased as possible, in the sense that we don't want to supervise the clustering processes ahead of time, because we don't know ahead of time what are the features or what are the dimensions that determine these classes, particularly talking about disease variation. And we can come back to that as we move forward with the examples that we'll read. And third, we want to be able to use, if you will, a divide and conquer strategy, or as some other people describe it, a lock and key strategy, to be able to combine the clusterings in multiple dimensions into a single class, into a single overlapping class. So the way we approach that process is by doing clustering in each individual dimension initially, doing an optimization of those clusters, usually with a dual objective optimization process that, on the one hand, optimizes the number of individuals in each class, and on the other hand, optimizes the identity of the individuals within each class. And then we cross or identify those individuals who share features across multiple different domains. So to give you a simple example, although I will give you much more detailed examples later, we can identify what are the individuals that share a certain number of features in the images in the brain. Then we identify the individuals that share a certain number of gene variations. And then we combine those two clusterings, bi-clusters, into sets that, or complex relations that identify those individuals that simultaneously, at the same time, share features on the imaging and gene variations. And you can do that across multiple domains and eventually label those classes depending on what are the features that are shared. Again, we'll hopefully clarify this as we move forward. And then you can identify, or rather separate those features and create new classifiers using those features. So that is a process that we developed initially with the name of PGMRA, but we can go back to that later if anybody cares to know. And use that to classify, for instance, personality traits based on, individuals based on the personality traits or the phenotype. We have used genotype classifications and imaging classifications, and then cross those domains to identify shared features between individuals or among individuals and create new classes. The first example that I'm going to give you looks at the imaging piece. This was work that was primarily done by Javier Arnedo, who's now in Spain as part of his doctoral thesis. And in this case, what we wanted to do was try to identify what features of the brain of patients with schizophrenia were shared by individuals, or by groups of individuals within the overall class of the schizophrenia. Now, why is this relevant? We can spend a long time talking about this, but one of the problems that the classification of mental illness in general has had, schizophrenia in particular, for the past 25 or 30 years, is that even though all of these persons express the same symptoms, when you try to identify either genes or imaging features or blood tests or any other biological marker that will allow you to consistently separate persons with the disease from normal people, you fail over and over and over again. And the hypothesis that we espoused when we started this work was the reason for that failure was that we were averaging globally instead of averaging locally, and we were not identifying the critical features that were shared by smaller groups of individuals, not by everybody with schizophrenia, but smaller groups of individuals that made them critically separate or different from other individuals with the same disease, as well as from individuals in the general population. With that in mind, we used the methodology that I was just referring to, and in this case, what we were looking at is at the anisotropy, as it is called, which is the level of organization, intrinsic geometric organization of white matter tracks within the brain. White matter tracks are the wires inside the brain, if you will, that connect different parts, one part of the brain to another. We were then identified the fiber tracks that were different in people with mental illness, in this case schizophrenia, and classify them into vectors for each individual and then the vectors into matrices. As I mentioned, we did non-negative matrix factorization for each of these individuals on multiple levels of, multiple number of classes, if you will. Then identify images defined by the bicluster of the individuals that share the feature and the feature, and then the multi-objective or multimodal optimization that I just referred to, optimizing for number of individuals within a class and identity, and then the hierarchical classification. So what happens when you do it? You identify biclusters. Biclusters are groups of individuals that, in this case, that share the same variation in the wires within the brain, or if you will, variation within the same wires within the brain, and that's how we use it. And then we took those clusters that were statistically different from the rest of them and compare the content of those white matter tracks, if you will, into those wires with either all of the other patients with the disease or with the controls. And as we predicted, what you find is that these individual clusters that identify well-defined groups of people that have the same abnormality are statistically different, both from other patients and from the controls. And you can, look at different examples of very tightly defined clusters, not so tightly defined clusters, but in all cases, we were able to identify groups of individuals that share very critical variations in the appearance of the wires within the brain that separate them from both other individuals with the disease and normal control. And when you look at that, you identify that there are specific changes inside the brain of these people that correlate with specific clinical changes, meaning specific symptoms and signs of disease. So these clusters that we were able to identify have medical meaning. They're not just random groups of fibers that are abnormal, but those groups of fibers that are abnormal overlap with, or if you will, jointly classify, have a relation, a statistically significant relation with clusters of symptoms that are also distinct. Incidentally, they have biological meaning. In other words, the parts of the brain that were abnormal in groups of individuals that had specific symptoms were related to those specific symptoms, if you will, were part of the biological processes that led to those specific symptoms, making it biologically meaningful. We expanded the same process to look at the genetics of the disease. As I mentioned to you, genetics of schizophrenia have been a minefield or a cemetery for good ideas, because every time that a group had thought that they had identified the gene that was different in people with schizophrenia from the general population, attempts to repeat that identification or that finding were unsuccessful. And again, the thought was here that the reason for that lack of success was that we were looking at global averages instead of trying to identify specific groups of individuals with the disease that had specific variations. And the approach is the same one more time. We did by clustering of genetic variations on one hand and of specific clinical symptoms on the other. This is a very large National Institutes of Health database called DBGAP. And then we cross them, or did the hypergeometric relationship between the two to identify those groups that were that shared simultaneously genetic variations and clinical symptoms. These are examples of those by clusters for the SNPs. So for the genetic variations, I mean, you have individuals that share a relatively large number of homogeneous variations in genetic variations, where you have others that are much more heterogeneous. But what's clinically important here is that when you relate using the hypergeometric, the variations in genes with the variations in clinical features you can predict who are the individuals who have specific types of disease. And these are actually examples of what do the clusters look like. We're not gonna, I don't really have the time to explain each of these slides in detail, but please make a note, and if you have questions, we can come back to them later. But for each group of SNPs, you have, average significance values for all the SNPs when you compare the frequency of that particular variation with the frequency in the general population. You have some that are very good, some that are very bad, and then you have the relationship of that specific set of variations with the risk of disease, meaning what's the likelihood that if you do have this particular set of variations, you will have the disease, schizophrenia in this case. And for some groups, it's very well defined, 100% for that, some not so well. In this case, you can have these variations and still have 17% of individuals who don't have any illness. Those genes are related to specific biological functions. And again, the biological functions that you identify using this process, using ontogenic analysis, are relevant for the process of the generation of symptoms of schizophrenia. And when you classify the combination of the two, meaning you identify those individuals who share specific genetic variations as well as specific symptoms of disease, you can identify small networks of non-overlapping groups of individuals, basically. In this case, it's the shared subjects looking only on the genetic variations. It's the gene subjects looking only on the symptoms. And you can tell that there are groups that are disjointed, are non-overlapping. All of these genetic variations are distinct from, say, these genetic variations, and these are again driven by individuals. Same for the symptoms. And when you cross them up, you identify, again, using the hypergeometric, you identify combinations of symptoms and genetic variations that are highly significantly different from other groups of individuals who have other genetic variations and other symptoms, as well as from the normal controls. So that was talking about schizophrenia. And I told you that we did that looking at the association between symptoms and brain features, and the association between symptoms and genetic variation. When we published those data, we had a rather, I wouldn't say surprising, but rather strong opposition from people who did traditional statistical genetics. Because the concern was that we were overfitting our functions and that we were identifying individuals that would be true for this sample, but would not be true for replication samples, even though we had actually done this in multiple replications. But that was the concern and that was the criticism. So we published a rather extensive analysis of the comparison between the traditional statistical methods and what we were doing using these non-negative matrix factorization approaches and clustering across multiple domains. And basically what this slide shows is how the method that we propose compares to standard traditional statistical methods that are simple contingency tables in which you count the frequency of an individual genetic variation in two sets of individuals. That's what's called GWAS, for those of you who are familiar with genetics. Again, it doesn't really matter so much what are the specific differences, but what we're able to show is that the methodology that we proposed had high reproducibility and statistically made sense. And it made sense not just because the way of identifying the lock and key or the hypergeometric probability of combination in two different domains was high, but because when we use the optimization of the clusters based on a good clustering algorithm, and by that we mean finding those clusters that have the highest probability of sharing the same features, but also the largest possible number of individuals, and allowing that for any one individual, that that person may belong to more than one cluster. And so allowing enough fuzziness to identify features that would assign a given individual to more than one possible class. By doing those two things, we were able to reduce the possibility of overfitting on the one hand and increase the reproducibility of the findings on the other. Using the same approach, we try to tackle the process of more common universal genetic variation that does not, or rather biological complexity, I should say, because it's both genetic variation and phenotypic expression that applies to everyone, which is temperament. For those of you who are not familiar with the concept, the temperament is that part of our personality that is hardwired, that comes from birth. You know, you have babies that cry more on the crib than others. You have babies that are brittle and easy to startle, some that are not. And that kind of very biologically driven process that leads to separate behavior in children and adults is what we call the temperament. On top of that, we tend to learn things and to modify our behavior on the basis of education, cultural norms, and so on. The component of our behavior that is overlaid on the temperament is what's called the character. So the combination of those two is what we call personality. We then approach the problem of trying to identify the genetics that underlie that type of behavioral variance in the human population, looking at first the genetics of temperament, then the genetics of character, and then combining the two and looking at features of overall personality that would be predictive of well-being, of feeling of health. So I'm going to quickly sort of give you the flavor of what this work looks like. We looked first at temperament because it's the more ingrained part of the process. We were able to identify subjects that shared specific gene variations initially this is the overall population. These are two example clusters. We were able to demonstrate that the individuals that do share those genetic variations tend to have specific temperament features that are distinct and different from individuals who are with other temperaments or with the overall population, just as we had in the case of schizophrenia or disease. Then we identified what are the functional roles of the variants that predict belonging to one of these specific temperament classes. And again, we were able to identify groups of individuals that form relatively disjoint networks, although not quite as disjoint, some of them are, not quite as disjoint in the case of disease, as you would expect, because it's the more common feature. I mean, all of us have a temperament. And then we were able to show that the belonging to the specific temperaments correlates with how healthy these persons are. So we identified 3 networks that correlated with these health indices, if you will, based both on the variation on the gene pool and the temperament measurements that we took. Again, the type of genes that are going to into creating or assigning participation to each of these classes makes sense from the point of view of what type of roles you would expect them to have, in the sense that most of them are regulatory or coding genes. And they tend to participate in processes within the biology of the cells and the brain that are, again, biologically consistent, if you will. They participate in processes such as regulation of stress and interpretation of emotional content of reality and things of that sort. And they have, again, very specific regulatory processes within the genetic makeup of the person. We look then at character. Remember, again, these are processes that are driven by learning primarily, and that depend on the life experiences of the person, not necessarily of the gene pool so directly. And we were able to identify, in this case, 5 different biclusters or genetic networks that predicted both the type of character that people will develop and that the combination of those 2 dimensions of information are predictive of how healthy these people are. Then we went on to combine the 2 at a higher level learning process. We did by clustering of both temperament and character, combined it and created personality profiles that were driven both by genetics and by behavioral features that allowed us to, again, identify very specifically groups of individuals who had personality traits that were common and that led to well-being, varying levels of well-being. I'm not going to spend too much time going through this in detail, but basically the upshot of all that is that we were able to identify 3 networks of relationships between behavior and genes that are related to personality features, self-awareness, emotional reactivity, and self-control that are predictive of well-being for that particular person. This is kind of a fun fact. We were able to move from that work forward and identify how those genes separate humans from Neanderthals, modern humans from Neanderthals and Neanderthals from chimpanzees in the genetic networks, such as those 3 networks that drive control of personality features are dramatically different as you move from chimpanzees to Homo sapiens. And that's what, if you will, makes us different. And the expression of those genes that are uniquely human is actually expressed in very specific parts of the brain that are related to those functions of self-awareness and emotional reactivity. So where do we go from here? We have done a fair amount of work in how to use these approaches to create classes of individuals. You could make the case of that in the context of disease, diagnostic classes that not only are predictive of outcomes in the illness, but that allow you to embrace the complexity and avoid the trap of global averaging and dilution of the features. But all of this work looks at static parameters. We have only been looking at established diagnosis, if you will, in the case of diagnosis, or established personality, and we know that neither of those 2 classes is really static. They change over time. So take the case of depression. A person with depression has symptoms today and maybe perfectly well 2, 3, 4 months from now. So how do you account for dynamic variations, for variability over time? Well, this is actually not a simple process. If you think of it from the perspective of classification, because you run again into the trap of the case of dimensionality. The more measurements you take, the more likely you are to have sparse information to narrow down the distributions and to run into overfitting. So how do you handle that? Well, I won't spend much time describing how we're doing this, but we are looking at approaches to try to understand how you can repeat the by-clustering process and identify the same classes of individuals using a variety of different tools, including decision trees and classification processes with which you are probably very well familiar, that include supported vector machines and things of that kind, that we can project over time and use to identify these trajectories. So hopefully I have given you a flavor of how we are approaching the problem of complexity, how we think it can be treated, not necessarily solved, but treated using deep learning techniques, and what are the limitations of that, particularly the limitations of dimensionality, and how we can go about addressing the problem of dynamic evolution of these variables in the next foreseeable future. That's what's coming in front of us. So I will be delighted to take questions with that. Thank you.
Thanks, Gabe. There are a few questions on the chat. Let's see. I think the first question was from Habil. Yes, Habil, do you want to ask?
Yeah, thanks for the talk, Gabriel. So in a couple of slides, you showed phenotype at the base level of the gene of the deep learning, and then you built up on that. When the phenotypes are kind of like the personality traits, or the phenotypes that are you don't know how to put them in the matrix. How do you put them in the matrix to get the first
You're asking me how you code? Okay, yeah, well, you're right. That's actually quite a complex decision to make, and I would be lying to you if I told you what this exact process is, because I don't do that. The coding, as you might imagine, is done by the programming people. But you're right, coding is actually a key component of all of this. But not only because you have to code it in one way or another, but because we have to code it in positive values. We cannot code negative here because we're using non-negative matrix factors. So generally speaking, what we do is we take any of these measurements, we normalize it and turn it into a positive. Basically, that would be the short description of the process. We turn everything into z-scores, then we positivize it, give it an absolute value, and we use those numbers to generate the vectors. Specifically in the case of personality, the measurement we have used is the so-called temperament and character inventory, or TCI, which is a very, very well established questionnaire survey to identify traits of personality along 7 dimensions, 4 temperament dimensions and 3 character dimensions. There are others, this is not the only one, but TCI has been around for about 25 years or 30 years, and it's been translated, culturally adapted and validated in a number of different environments, including Arabic countries, Chinese, Quechua, in South America, all sorts of places. It's translated in about 50 languages. And there are validation parameters of the structure of the personality using this instrument in a number of different settings. So we were pretty confident that we could use it. And in fact, the data that I reached through, I didn't really show you, but I reached through, were confirmed in 3 different samples, one of American individuals, one of Korean individuals, and one of German individuals. So,
is it a discrete or continuous trait?
No, the questions are discrete. It's a 234 question interview, but the traits are dimensions. So there are dimensional traits. Okay. You have to discretize them when you code it, obviously, but the traits themselves are dimensional. That's why I was telling you, we use the normalized values and then we discretize them. Yeah.
Thank you.
The next question is from Matthew. Matthew, do you want to ask?
Sure. Yeah. I didn't hear you say you were using k-means clusters, but it seems that that's what it is. I just wanted to make sure I had that right. Are these multiple k-means clusters, and that's part of the approach being used?
No, we use NMF. Actually, it's not k-means.
NMA.
Yeah.
Okay.
The k-means I mentioned at the end because we do use k-means sometimes for the post-hoc classifiers. Once we have the traits identified, when you do a supervised classifier afterwards, sometimes we use k-means. And that's for the longitudinal work. But for the initial description of the by clusters, we use NMF.
Okay, thank you.
Thanks. The next question is from John Parsi. John, would you like to ask?
If you don't mind, you can read it.
Okay, sure. So the question is, knowing that the DNA nucleotides under investigation are a germline and every individual with a genotype of temperament could be labeled as its phenotypic expression, How do we safeguard individuals from agencies, government, not to abuse this research to stereotypically stigmatize an individual, or worse, a family with the same DND SNP?
Good grief. I have no idea how to answer that. All I can tell you is that what we have is de-identified data. So we don't know who the person who has this information is. Whether this would be amenable to manipulation or not, the answer is yes. But what information that we generate cannot be manipulated. Ask Google, Netflix, Amazon. Everything we do gets manipulated for commercial or other less pleasant reasons. We take the safeguards that biological or medical research needs to take and must take on every level, and we try to stay away from knowing who these people are. But you're right. Anything we do can be used for ill purposes, and it's a conundrum that the science field as a whole should take, not necessarily research on temperament and character alone. I think that the fact that, for instance, that the fact that any browser maintains record of 14 clicks at any given time, and that's, we know, is being used regularly to develop your pattern of behavior. That's essentially a measurement of temperament and character. Tracking what you choose when you are browsing through a website and using that information to sell you stuff, which is what all of these companies do, is essentially a way of manipulating your personality and personal preferences. Whether you add genetic information to that or not is somewhat icing on the cake, if you will. It doesn't change the substance of the problem. You're right.
Yeah, I'm as a scientist and an engineer, I'm trying to intrigue our colleagues that I mean, there are so many things that we can do, but the point is here that we should think about it, how to safeguard that against others that they can abuse it. But your presentations and the whole process was so beautiful. Thank you so much for honoring us and coming today.
John and Gabe. Other questions, feel free to type on the chat window, or you can raise your hand.
I will add one more thing. So on Facebook, for example, there are a lot of very friendly users who are going to ask you questions that are actually the secret questions that you have for all systems where you set up your authentication. Probably the same kind of users on Facebook are collecting this type of information about all of us. So don't answer questions that are out there.
One of the questions I had, Gabe, I think you talked a little bit about deep relational networks, right? I don't think you kind of touched upon how you're using it in your models today. But in general, I think one of the challenges with this complexity or the dimensionality curse you're talking about, I think is even like how you're processing this information on the systems you have, like what are the storage challenges you are facing? Some of these things might be interesting for the AI team in here.
Sure. Well, a couple of things. First, on the deep learning piece, we didn't talk about that, but I did show you one slide in which I talk about the post-hoc processing. You have to interpret all this information. You generate by clusters, you generate high-level relationships between multiple by clusters, and then what? What do they mean? So that's where the additional knowledge-based learning comes in, and where we use things such as natural language processing, interpretation of ontogeny, that kind of thing that requires expert knowledge, and that it's added on to the by clustering process, which is not particularly informative if you don't have somebody looking at it. So you do need expert knowledge there, and post-hoc interpretation. Regarding the challenges of processing, these days is relatively less. I do have a slide I thought about putting because I figure it might be interesting to you all, in which we have different data set sizes as well as different number of features for each of those data sets and the processing time in different clusters. We have smaller and larger clusters. And yes, of course, it can, depending on the size of the data set as well as on the size of the number of features that you're looking at, you're talking about days of processing.
Right.
So for instance, even in large clusters. So for instance, when you look at, which I didn't share with you all today, you look at deep brain stimulation prediction based on imaging features. So you're talking about whole volumes for a relatively large number of individuals. It can take 3 or 4 or 5 days of processing for a single run in a large cluster. And that's just for the biclustering. So when you look at relations, it can take longer. So we use what everybody uses. We use clusters that are university-based. So we typically use the cluster at Washington University or the cluster based at the Cancer Center in Granada.
Okay.
But storage is not that much of an issue because storage is, most of these things can be, once you reduce them to matrices, that can be stored in your local hard disk. So that the volumes are more of a problem, of course, and for that we typically have those in the servers at the university as well. But again, it's relatively a smaller problem because we're not using big data sources. We're using patient data sources. So the numbers in the upshot are a couple of thousand, and usually are fewer. For volumes, for instance, we're usually using fewer in the hundreds. And genetic data sets, once they're coded, they're small. It doesn't matter how big they are. They're in terms of actual information demand, they're relatively small. The biggest issue is processing, and we rely on large clusters. We have not done any of this work, although we did think, and in fact, I have a CUDA device that we plan at some point to use for this, using NVIDIA cards to process this type of information. We never developed that to any, to any meaningful extent. I think that will be more efficient. So eventually I'd like to do that, but it takes a lot of programming and we haven't actually had the resources to do it.
Yeah, and I think you shouldn't be spending your time and resources on that. You have all these other research aspects that you need to look at. And that's where I think matrix researchers could help you with all our clusters and compute resources and programming capabilities.
I'd love to talk more about that with you.
Sure, sure. There is one more question from Edmund. Do you think using dimensionality reduction methods such as autoencoder and VAE would help the dimensionality curves? I know it would affect the explainability of features compared to raw features.
Well, right. I mean, you saw we do use autoencoder. So the answer is yes, obviously. I don't like the idea of using standard data, although I have standard data reduction strategies such as PCA or ICA, because those are supervised usually. And we try to stay away from supervision for obvious reasons. We don't know what we're looking at. So if we start with the example of the 3 colors, of course, there you can use ICA because you know who's who, but if you're starting from the other end, it's impossible to do that. So, and make any sense of it. So we do use autoencoder based on NMF. We don't use data reductions that are supervised. Not initially anyway. We might use supervision or partial supervision at the other end when we are making interpretation.
Thank you.
And let me just complete the sentence. We do use supervision at the end, because we're supervising on the basis of the cluster, so we identified with the autoencoder, if that makes sense. So we identify the big clusters and the relations first, and then we supervise on an ongoing basis on the basis of those classes that we identify with the autoencoder. I hope that makes sense.
Yeah, thank you. Don't have any other questions. Thank you so much, Gabe, for your time today. That was very insightful talk, and I hope it opens up some new ideas within our matrix community, and hope we could help and support you in some of your computational needs as well.
Thank you very much. It's been a pleasure to be with you.
Yeah, bye.